Well, good morning again and welcome back. I thought we were going to be 10, 11 people in this session, but no. We have big fans of translational nanomedicine, so thank you for being here. It's going to be, let's say, a journey, different kinds of initiatives. Normally, it's a pure ETPN session. By this, I mean that we only show things that we do. And we've decided to put, let's say, a highlight on things that are done locally and to give the words also to, to UK partners, working to foster the translation of nanomedicine towards the clinic. So I'm super happy that we changed a little bit this session. And that's exactly the spirit of this NME conference. So 3 talks today. The first one will be by Angel. Hi, Angel. Del Pozzo, who's doing many things, but who's also the responsible of the SAFE and MedTech OETB, Open Innovation Testbed. Who doesn't have any clue what an Open Innovation Testbed is? Don't feel shy. Raise your hand. 2 of them, 3, and then 20, be honest. You do know what it... No, okay. So, Angel, you've been giving several talks at ETPN events towards this journey of Open Innovation Testbed. But today is quite different, because today we are going to talk about some real case, real life study, like how these kind of European big projects like Open Innovation Testbed can help companies to grow in the field of, let's say, health technologies, nanomed. And we are super happy that you give this real life example, and you have 25 minutes plus question. You have a pointer, you have a presentation, you have everything you need.
25.
25.
Okay.
Not 35, right?
Yeah.
I know
I
you.
can go from 10,
I know.
From 10 to 1 hour.
No, please. And Angel, the floor is yours. Absolutely. Number 2, right? Yeah.
And to move the slides.
You have the pointer?
Okay. First of all, thanks a lot for being here, because as Alex said, I expected really few people, because in the other room there is that really impressive Maria Jose Alonso. Professor, don't go now. You are now here, and you have to stay here for the time. But yeah, they counter program my talk with her, and I was afraid. So really, thanks a lot for being here. As Alex said, I am going to speak about the Open Innovation Testbed, how they can help in translation of nano-enabled medical technologies into the clinics, and afterwards into the real market. That is what this is made for. So I will start by explaining this is a European project. It is funded by the European Commission. For the official presentation, I will have to put the project number and so on. But yeah, this is the European Commission decided to fund a new tool, let's say, a new instrument that are the Open Innovation Testbeds. They started funding this 5 years ago, and up to now they have funded 27 Open Innovation Testbeds with around 300 million in total. And they decided to do this because they identified a need this need is every time there. All of you that are working in research and innovation, you know it. There is a clear gap in advanced technologies when you have to move from your results, your prototypes, your TRLs, let's say 3, 4, into more advanced TRL and going afterwards into the markets. So they chose some sectors quite different from biomaterials, flexible materials, building envelopes, many different things, batteries, I think, many different things that they had the feeling that were in need of having this support from these OITBs could help these innovative technologies becoming into products and moving into the market. And into this endeavor, they funded 3 Open Innovation Testbeds in medical technologies. One is SAFE and MedTech, Open Innovation Testbed. The other 2 are TVMed and MDOT. In our case, as we are members of the ETPN, we have worked for many years on medical technologies, on the application of nanomedicine and nano-enabled medical technologies. We decided to choose as our scope, let's say, because this was open to any kind of medical technology, and a medical technology is from a small bandage to a X-ray machine that costs 20 million euros. everything in between is a medical technology, a medical device. So we decided to focus on, we had to reduce the scope, and we decided to focus into nano-enabled medical technologies, okay? Any medical technology that is making use of nanotechnology to work. So our aim is into this overall framework of medical technologies, of open innovation testbeds, sorry. Our aim is to contribute to this full deployment of the potential of these innovative medical technologies based on nanotech. So what we are doing, we are creating an innovative open access platform to offer a group of capabilities, know-how, our network and our services in order to, as I said, help in the development of such technologies by incorporating intelligence at the beginning, testing, assessment, upscaling, market exploitation, clinical validation. I mean, this is, you can see this like a bundle of services and know-how, I would say in the other way. First, know-how and knowledge, and afterwards services that we can offer to any developer in this field of nano-enabled medical technologies to go ahead into the development. You see there probably some familiar faces that are already in the room, others that are not, but are quite known in this community. This was from our previous General Assembly in Braga. So, because we are 28 partners from 3 continents, so good doing this again? Never good doing this again with so many partners. No, I mean, I enjoy it, and all of them are meaningful and are needed. But it was really challenging to manage so many people and so many profiles. But you see that we have, I could say, the dream team on nanomedicine, nanotechnology, and so on, from characterization to physicochemical assays, to in vitro, in vivo, quality, regulatory, health technology assessment, clinical application. So these are the people that will be the embryo, let's say, for our future, that would be to be an entity into the market, an open, the Commission likes the wording, single entry point, unique stop shop, unique window, this kind of thing. So the idea is to integrate all these services and all this knowledge into one single entry point where you can go with your technology and you can get the support of all this pool of experts, of all these services, equipments, and people into your development. I mean, of course, now as we are a project, we are walking into supported by this funding, but after the project ends in September, that is quite close, we will be a commercial entity in the market, competing with the usual CROs, the usual regulatory consultants, and so on. But our idea is that this integration of knowledge and services will be quite unique and will really help in the development of the technologies. How we are planning to work? You see there what we call the YTB service process. This is how we build the research project on one hand. So you see there that several work packages are, we have like 12 work packages because, as I say, the project is huge. It's 18 million budget and they mentioned 28 partners. So you see there how we see the project, but more important, how we see how we are going to work into the future. My idea for today, because I explained this other times, as Alex mentioned, of course, there is many fresh people here that I really appreciate. But my idea is to show you how one company that is part of the project, RCL, that is a company based here in the UK, they pass through all this process, let's say, and how their project, they are partners of the project, of course, but how they benefit from all this approach into moving their projects and their technology for translation into the clinics. This may serve also for some of you that are already in a research phase, in a TRL 3, 4, something like that, to really see and understand, because this session is also about that, about hurdles on translation and how to overcome them. You will always see how many things you have to do with a product, in this case a nano-enabled medical device, in order to get into the clinics and afterwards into the market. Because of course when we do research, I've been there in the bench, playing, with the chemists and so on. It was dangerous because I am not so good with my hands, so I decided to move into this dark side of the management and so on. But I've been there, and you can have a fancy technology, really good, really promising, but sometimes we as technologists, let's say, we are not fully aware of all the needs that we are going to have to fulfill in order to move into the clinics and patients. So our vision is that we have like two levels, okay, of support. One is the technology, what we call the technology assessment area. That is, let's say, the entry point for a technology where somebody like you is coming and with some promising idea, with a kind of, as I said, something like a TRL3. And from that point, you can be an SME, an RTD, even hospital, even a company. So you come into our system, let's say, through some questionnaires, some interviews, also the application of some tools like a decision support system in silico that is being developed here in Liverpool by Neil and the group, and so on. Our experts should be able to offer you two main outputs. One is first evaluation of the cost effectiveness and reliability. I mean, you think that your technology is the best one in the world, of course. Is your child, is your baby, you are working on that for 10 years. But maybe sometimes this is not easy to scale up, easy to apply in the future. And something that makes Safe MedTech a little bit different, maybe it's not needed by the system. You see there the words HTA. That means Health Technology Assessment. We have translated this into early dialogue, early advice from experts in this field. What is this meaning? All of you are aware of the regulatory concerns, the regulatory hurdles, the regulatory barriers, that is to get your CE mark and have your product, or your FDA, but of course we have to foster a European adoption of technology. So you have your CE mark, you are super happy because your product now can go into the market. But the next step is that you have to go to the healthcare systems, even public or private, and somebody has to identify the need and adopt your technology and pay you back for the technology. I mean, when you produce a car, you need some certifications here and there, but you go to the market, to a free market, and you sell your car. But with drugs and health technology, it's not the case. So you need to understand what the healthcare systems are going to ask you, your product, in terms of performance, to be adopted. Discussing with the experts on this, they put an example. They had, in a real hospital system in the Basque Country, where I come from in Spain, they had a routine analysis, I don't remember for what, that was already established, already optimized, already had an agreement to buy all the consumables and so on with the company. And somebody came from the Basque Country, I mean, well appreciated, the company from there, with a really new, outstanding technique to get into picometers of something, whatever they were looking for. And it was already CE mark approved. And the system was looking at that and said, okay, it's fantastic technology, it's amazing, it's super good, but we don't really need that precision in the analysis. I mean, maybe you have to focus in other application, I don't know, drug detection or things like that, but we don't need this, because with the microgram, we are more than good. So you are going into the picogram, but if you have asked before if somebody is needing this, probably you were deciding how to readapt or if it is meaningful to develop your technology. This is one part, from the regulatory and health technology assessment point of view, what they think about. Of course, it's not mandatory, it's not prescription, it's just an informed opinion, let's say. And on the other hand, we will propose what we call a technology assistance proposal. What is next? Depending on where you are, on your degree of development, on your TRL, which program should you follow to continue developing your technology? And you see there that we incorporate all that possibilities on, okay, you see there on, I knew that I was going to kill the presentation. Okay, you see it there. And you see that the arrows are in parallel because you can enter into this technological assistance area at different steps. Maybe you have already a huge characterization package that you already developed and you are happy, and of course the experts will evaluate the data and will come back to you with an opinion on if you have enough, you don't have to repeat, okay? So as you see, it is in parallel because you can enter through any of the points. So, the nice part of the story for today, how is all this applied in a real translation challenge? Okay. We have a company, as I mentioned, that is RCL, Resonance Circuits Limited, that is here from the UK. We started a cooperation with them, I would say maybe 8 years ago, because we were part of a previous European project that was called No Cancer, that was coordinated by Indea Nanociencia, that Mark is there in the front line. And in that project, they were advancing their nanomaterial with some good results in terms of in vitro, in vivo, etc. But we incorporated them into SafeMedTech to go a step beyond, okay? So what they are doing? They are bringing their system to the YTV. I mean, this is like a role play, because I say it again, they are funded by the European Commission. Our challenge is to find real clients after the funding is ending in September. But this is nice to see how we applied our methodology to a real case. What are they doing? They have an injectable agent that are magnetic dextran nanoparticles that you put into a tumor, okay? You locally inject this there. And afterwards, with the RCL generator, you create a magnetic field, so the particles are heating. Probably you know this concept from other companies like MagForce. And you are creating this heating of the tumor. You can go for the thermoablation of the tumor, try to really destroy it with the heating. This is challenging because you can create a lot of cytokines storm there, and you can even release cancer stem cells around. Another possibility, that is the one that was more studied for this technology in our case, is to try to use this as a sensitizer to, let's say, create some weakness in the tumor and make it more suitable to be attacked by the chemotherapy. So you see there what you can see from the eye. That's the vial where the nanoparticles are inside. As I said it again, they are multi-core magnetic nanoparticles covered by dextran. This is what you inject into the tumor, and you apply the heat afterwards. So what we did is what I mentioned first. We went into we met them, we passed them the questionnaires and so on, and we incorporated like 3 levels of knowledge. One is regulatory guidance from the beginning, what I mentioned on the health technology assessment, to get to the best indication for the technology in terms of type of cancer, and to create a development proposal, as I mentioned. Here was implied the people from the STA, from the Basque Country, Euroscan, that is an international network for HTA, Maraca International, that is the consultancy for regulatory, BioCaralty, my institution as the coordinator of this case, and the company themselves. The idea is to move from TRL5, that we consider that was the technology at that moment, into more like a TRL7 or 8. So what happened? With this first approach, as I mentioned, we integrated this discussion based on regulatory and HTA in order to define the best indication, because they were, in the previous project, in No Cancer, we were working in pancreatic cancer, okay? And now we were looking for a new application. So here we have like 2 developments, one for the pancreatic cancer to complete it in the clinical phase, and one new indication. They have some options, but this group, taking into account all the inputs that I mentioned, they decided to go into the prostate cancer as the next application. Okay. From that, as I mentioned, we define the technology assistance proposal. We put the questionnaires. We do the interviews with the company. They create like what we call a wish list, what they think that should be done, but it has to be counterbalanced by the experts. We get to this draft technology assistance proposals, also informed by the regulatory and STA, the decision support system, that is kind of automatized system to to classify the technology and some of the steps to be done in the characterization and forward on. Experts review, and we get to the final technology assistant proposal, and of course there is a continuous feedback and refinement. So the idea is we were there, you see, in the regulatory guidance and so on, and now from this technology assistant proposal we have an integrated program that includes preclinical, toxicology, biodistribution, protein denaturation, reactogenicity, and at the end, clinical research. Supported by SINTEF, Cyber, Trinity, Valdebron, INL, and the Institute of Physical Science, Ilie Murgulescu in Romania, in Bucharest, of course, the University of Liverpool, as I mentioned, and we even went a mile beyond and tried to also identify the risk for occupational safety. The idea, as I mentioned, is to move from this TRL 5 into the TRL 7 or 8. There you see who was participating, but the important thing, as I said, is that we are not, you as a client, you don't have to go to someone to do the characterization, to someone to do the in vitro, someone to do the in vivo. Try to connect all the results into one package, try to understand what is needed, what was done, what has to be repeated because somebody was not doing in the right way. The idea is that this integration of knowledge and services should lead to shorter time and less cost for the companies to the development. I will not go really in much detail about each step, but just because also the company didn't want to, to disclose everything, of course, they are into the market. But you see there, we made also for you to take note of the things, or part of the things that you have to do. The biocompatibility testing regarding skin irritation, cellular mutagenicity, leachables and extractables, of course, what happens with the particles if we put them into the tumor but they are released somewhere else. Also, in the occupational safety, how are we working with these nanoparticles at the production side, at the clinical side, and the safety for the patients? We have developed a risk assessment tool that integrates all the risk evaluation around all those steps of the development. Sterility, of course, quite important with endotoxin and so on. And next steps, we are really getting to the end, that is the formal records or reports. Biodistribution studies, of course, quite important to show if the particles are being kept, let's say, into the tumor or if they are released anywhere out. Of course, as I mentioned, the University of Liverpool has developed a full package of in silico evaluation of this PBPK based on these results, comparing the in vivo results that we are obtaining with the models that Neil and the team are developing here. And that is knowledge that we think that for the future cases into the Open Innovation Testbed may help us in the reduction of animal testing and so on. Protein denaturation, quite important. This is a complement activation being done, as I said, by Esperanta in Romania and also supported by Trinity College. Imaging to fully identify the characteristics of the particles with the most advanced, TEM, cryo-TEM, MRI, and so on. Preclinical, of course, this is quite important for as I said, there are two applications: pancreatic cancer that is already in the clinical stages, and the new application in prostate cancer. Of course, we needed to do a new package. We could reuse some results, and this is also good learning from the previous application for pancreatic cancer, but we needed for the prostate cancer a new model to show to the clinicians to be approved and the authorities to be approved for a new clinical trial. In this case, we had to use beagle dogs because it looks like they are the best model for prostate applications. So we are intensively working in that way. And the clinical study, as I mentioned to you, in a previous project called No Cancer, we were able to get to start the pancreatic cancer clinical application. But this is a problem that we all have in our research projects. The funding ended and the clinical study was starting. So what to do? As we were part of both projects, we were able to bridge between them, let's say. We agreed with the coordinator of No Cancer in adopting the clinical trial also as an important input for the SIFMedTech, and we have been able to continue with more patients. The results are, you see, that person there is a real person. You see what is on her chest is the magnetic field applicator, let's say. So she has already received by endoscopy the nanoparticles into the tumor, and now the equipment, let's say, is creating that heating, let's say, of the nanoparticles. What we have demonstrated is safety, because this clinical evaluation was not trying to go beyond that. But it's true that there is a common effect from the first 4 patients that have shown that they are having less pain. We are now trying to identify why, if it is because the tumor is being reduced, if it is because the heating is creating some effect there. But it looks like I mean, first of all, and most important, there are not side effects. Everything looks like safe. That was the main idea for the study. And they, of course, are planning now to expand it at least until 15 patients. And of course, we are also discussing the new prostate cancer clinical study to be done. One important step beyond that we made with the Safe and MedTech, with the adoption of the pancreatic cancer test case, is that many people with pancreatic cancer, they have a metal biliary stent. It's routine practice. In Europe, out of Europe looks like they are more plastic, but in Europe, or at least in Spain, they are metal. So of course you can easily understand, if we have magnetic fields and metal, it sounds like something at least to be explored. And we could confirm with some experiments that, and this was not creating any safety issue. And the Spanish Medicines Agency, the regulator in Spain, they approved that we can include these persons with metal biliary stent into the study. Pancreatic cancer, you know, is quite severe cancer, really difficult to manage, and there are not many patients that are eligible for this type of studies because their life expectancy is not so huge. But now we can incorporate more due to this new study. Of course, discussing with your urologist into the prostate cancer. We are also making some reactogenicity study supported by Trinity College, and complement and all these things, and of course the regulatory review with our experts in order to ensure that everything is ready for the CE marking. This is what happened with RCL. Nice story to tell, because you see in human application of nanomedicine, let's say, that is not so common, or was not so common until the last years. And what is the future for us? As I mentioned, we will be an actor in the market. So we created an entity that is called the OITB pathway, to try to take, if the Commission is doing marketing of OITB, but why not? If you look into the internet for oitb.eu, is our website. And the idea is to develop what I mentioned. Do what is being done into a research and innovation project, an innovation action to be exact, is to move this into the market. So we already created this entity that is a non-for-profit association, so we can accommodate much of our partners into that. And of course, the idea is what I mentioned, to offer this early advice, to combine the technologies according to the TRL, and to offer this development pathway. We see ourselves as an intermediator between the needs and the ideas. And as you see, we will have these external services for technology developers. That is mostly what I mentioned in the presentation. You see all there: clinical, STA, quality, business, prototyping, production, regulatory, characterization, in vitro, in vivo, and so on and so forth. What can I say here? Just two last words, because Alex is controlling me there. Yeah, this could be good for you in two senses. You can approach us if you have something under development and would like to. We made some open calls, but they are now already, of course, as we are ending, they are closed, and we are working with other companies. But if you want to make use of this, you can approach us. We can try to look for the funding altogether. And also, on the other side of the story, of course, this is not a closed club. As I mentioned, it's a non-for-profit association under the Belgian law. So if you are, also as RTOs, you are providing services in the field, you can also be part of the other part of the story. Try to be part of our pool of service providers and get also more business opportunities for you. So there are these two ways where you can be interested in knowing more about the OITB pathway scheme. And that's it. I think that it was on time. Really happy to answer any question, okay?
I thought you don't want me to speak. Okay, already question.
Hi, and thank you so much, Angel. I mean, it's Angel.
As you prefer, no?
Okay. Say NMT or something. It is extraordinary, incredible. Thank you so much for participating in that or organizing this. It is replacing the UNCL and sorry for this question. Sorry, I'm old. I mean, I haven't been there.
Very accurate question.
So if not replacing, I mean, is it something upgraded, new version 2.0, trying to help all the technologies and non-technologies and in Europe? And I have two more questions.
Okay, no, on this one, every year I present a new update on the CFMTech, and in Braga, 1 year ago, I was connecting between this and the Translational Hub, Nanomedicine Translation Hub, something like that.
Yeah.
That was the roadmap that we all, as a community, we identified maybe 10 years ago. And if you see, it's not far from that idea, because it's trying to connect capabilities, trying to integrate what is needed for this. So more than the UNCL, I could say that this is inspired, but that's because it's more than characterization. Of course, we have integrated some protocols and ideas from the UNCL, because we have there Trinity College, SINTEF, people that was basic for that. University of Liverpool, that were basic for the UNCL. I was not part of that, so I only have that names on my mind. So it is. I mean, we are not going to have more funding after September, but we will try to see if we can make a business out of this. Okay.
Just maybe a word for those of you who doesn't have a clue what the EUNCL is. It used to be a European infrastructure inspired by the ETPN as part of the global set of tools that we used to call the Nanomedicine Hub. We've identified three main hurdles. Mentoring, to launch like a spin-off company from industry, like you have access to knowledge. Then characterization, we had the EUNCL. It was like free of charge, complete preclinical assessment of the quality of your product, let's say characterization, even to in vivo assays, right? And it was for free, and it was funded by a European project. So it was extraordinary, and it works really well. So they stopped the funding, and as you do. And finally, there were the pilot lines for GMP manufacturing, going to milligram to grams or gram to kilograms. And these have been transferred to OETBs, which are somehow, let's say, matching the whole process. So it's not exactly the same, some kind of continuation. Yeah. And since it works, we end the funding, right? As you do. Can I ask just a quick question? Exactly like, do you have a business model that works? Because so many services, so many partners, so many expertise. How do you transform that in something which is sustainable?
Yeah, we are in that moment now working on that. I mean, we anticipate to have some agreement with the service providers that will pay kind of a fee for being part of this, and also under discussion on kind of percentage of revenue to be put into the pot. But all of them will be also part of the association, let's say. It's the idea, because why we made a non-for-profit. I mean, the 27 Open Innovation Test Beds, they have the same challenge now, because some of them are we were at Europe Forum last week in Sweden, so it has been a nice journey during this June. And we had a workshop on our common challenges and common future vision, because we have to build that sustainability. So on one part, we think that the providers can get advantage of this and also contribute to the sustainability. And of course, we have to find clients on the industry that are the real goal of this. If we are not needed, the market will spell out. us from the market.
On Thursday, we have a session on European projects in nanomedicine, during which you have another open innovation testbed, which is called Phoenix. You have a presentation, a shorter one. So I encourage to come there, let's say, closer to the beginning. So you have different level of maturity, different expertise. But I'm looking forward to find solution, and I hope that the Commission will help us also on the legal point of view to make this a running business.
Yeah,
because that was part of the main objective at the beginning, and you did deliver some fantastic outputs.
Yeah, we are also working with the Commission quite close. They are now doing a report on the performance of the OITBs. And I cannot speak for the community, but I am one of the fosters of this integration of the OITBs. I can say, and I think that is a common view of the OITBs, that we don't want more money for us. Of course, if they want, it would be much appreciated. But that they give money to the companies and incorporate these aspects into the EIC, EIT.
Or in proposals, or even in proposals. They are supposed to be translational.
Yeah, of course.
Sometimes they ask for an HTA now in proposals.
Yeah. We will be eligible for any proposal. So, of course, if you and we are not bad in writing proposals.
So I guess as UK companies, startups, spin-out will be eligible to apply for support.
No, but
to apply, yes.
To apply to get it, it's
yeah,
another story.
But we will be a commercial service from that
amazing.
moment on. So if anyone comes with the money, of course, we can take pounds. I think.
Of course,
but not. Through the the European Innovation Council, the EIT, we would like that they put a special mark on the programs, like not forcing, of course, but suggesting that people put budget into the proposals to get this kind of services.
And not to reinvent the wheel every time to start
No.
from scratch.
And
last last
please.
question. What is nanomedicine TRL5 according to you?
The tricky one.
Yeah. Okay. No, we are used. I will give the ball back to Alex, because when we prepare proposals for nanomedicine, we always use a nice picture that was released in one of the roadmaps of the ATPN, that is connecting TRLs with nanomedicine. It's true that non-nanomedicine in health in general, in health research, it's not so easy to identify these TRLs. But for me, TRL5 would be something, let's say, on the in vivo, let's say. And TRL7 would be the first clinical application. More or less, I think that for me, it is there, okay?
GLP in vivo?
The GLP could be the 6, I could say. Because they say, like, demonstrated in, I don't know what, real environment or something like that, so that could be. But it's true that we have to play as health community with these TRLs that are for, I think
I'm not matching exactly the purpose.
No.
In medicine, it's much more complex product. Yeah. So you can be in TRL for some part and the other TRL for other part of the solution. There's a fantastic article by Mike Eaton, one of the health tech tab experts that you do know well because you were an applicant, satisfied applicant, I guess. And a fantastic article on the fact that TRL doesn't match to nanomedicine easily. I'll send you the reference.
I think that originally TRLs were for sending a rocket to the moon. They are from NASA. And now the European Commission decided, wow, this is fancy, and applied it everywhere. So,
and let's change that in 5 years.
Is it being recorded? Or?
Absolutely. It's live stream. Thank you, Angel, once again.
Thank you.
Maybe we'll move to the next speaker and we have more questions at the end. We can take one. Oh, Robert, you had one?
Well, yes. Just comment question. You were saying you're not asking for money for yourself. But I think it can only work if there is some sort of coordination. You're saying there should be budget for this kind of analysis, but where do they have to go? There needs to be an entry point where they can go and someone to tell them, okay, you need this part of your budget for that, this for that, this for that. And otherwise, your business model is a bit weak, I think. So I think you should not be too modest, and also ask for money for the entry point. Let's say. Otherwise, there may be nice rules for having proposals with this kind of budget in there, but it will be hard to manage.
Indeed.
Yeah, no, you're absolutely true and right. And the question there is that they are putting us in front of those 300 million for the whole OITBs. and now it's like challenging us. But we are there to help the others to overcome the valley of death. But first we have to overcome our own valley of death. I used to tell this to the officers and the policy officers and so on. So we have to find a way. If they don't want to waste the 300 million, we maybe need some more, maybe coordination and support action to find integration between the OITBs. We are also working on that with the other two open innovation testbeds for medical devices in trying to, I couldn't say integrate, but maybe how to share services, how to share costs, because 3 single entry points, I mean, it is by definition single means single, not.
And we
so
we don't have time to speak about the industry competition which starts to exist.
Yeah, of course.
We
CROs, laboratories.
Let's see in 5 years where we are.
Yeah, but okay, we can meet in the coffee break or in the lunch, and if you want any other input, I will be more than happy, okay?
Thank you so much, Angel. Impressive. Thank you again.
Thank you.
Now, we welcome on stage Dr. Juliana Agerti from the CPI Center of Excellence on LNP and intracellular drug delivery. So basically, we will see how UK deals with it and how do you build the capacity in UK to make it go faster and better. Thank you. The floor is yours, Juliana.
Okay, thank you very much. So most people call me Jules Haggerty. And I think once we go through, you will see quite a lot of parallels with what's happening in the European ecosystem and what we're trying to do in the UK. And that's not a coincidence. So CPI have been part of many European projects, including open innovation test beds. And so there's a certain level of inspiration, I think, that's been taken from that model, because there's not really a clear need to kind of reinvent how we do this. So what it might be quite useful to introduce first of all is what a catapult centre is. So both CPI and the Medicines Discovery Catapult, who are one of our partners in this initiative, are part of a network of UK centres that were established to support the translation of high-quality research happening within the UK into commercialised products, services, with the aim of having economic and societal benefit. So it's all about translation, and it covers a whole range of different areas. But the area we're talking about today is going to be nanomedicine and technologies to support intracellular drug delivery. So CPI have actually been working in the nanomedicine area since, around 2014, 2015. The very first thing that I had to do when I joined CPI was write a Horizon 2020 bid for pilot lines for a gold core nanoparticle. And from that journey, from that success, we've started working with polymeric, lipid, inorganic, metal-based nanoparticles. But understandably, the focus changed a little bit to lipid nanoparticles around 2020. And so we've built a large group and set of facilities in that space. But this initiative today came out of the work we were doing during the pandemic, where it became clear suddenly there was a lot more interest in government and funders about these delivery vehicles. And they said, will you look at this area and go, what capabilities have we already got? Where are the gaps? What do we need to fund to ensure that we're solving some of these key challenges and we're making the most of the opportunity, not just in vaccines, but for future medicines? So this is what we came up with. It took a couple of years, basically, of refining and working with different government departments. But we received the funding at the start of this year. So that's what I'm going to be talking about. So what is the Intracellular Drug Delivery Centre? So it's a multi-partner program bringing together expertise in universities as well as 2 catapult centres into one centre of excellence. And you can see the partners down there, so I'll go into more detail about who's involved on a later slide, but you should see something familiar with open innovation test beds. So it's this idea of a single entry point or an easier access. How do you simplify access both for academics and companies, large and small, to those centres of expertise across the UK? We don't want to stop duplicating that expertise. It's about building it up, making sure they've got access to state-of-the-art capabilities, and making sure that we're becoming more than the sum of our parts. So CPI's particular expertise is around manufacturing. So we're very good at taking a process from an academic that works very well at a small scale, making it robust, scalable, ready to go into a GMP environment with appropriate characterization methods, maybe applying digitalization techniques as well and advanced modeling. But we've brought together the other partners so we can also think about design, characterization, looking at what happens so once it gets in the body, because we all know just measuring the physicochemical properties of a nanomedicine or a nanoparticle doesn't tell you what it's going to do when you actually put it in a patient. So we've deliberately brought together a consortium that helps us move across that whole kind of level of characterization. And we've put together some R&D programs to solve challenges and make sure that we've got a flexible industry and academic engagement model. So the mission here is providing, and there's keywords here, it's about, yes, infrastructure. So we've bought equipment and we have facilities, but we're trying not to just spend all of the money on new buildings, but also about capability and know-how, which will be there to support companies and academics. The focus initially is on delivery vehicles to enable RNA medicines, and that's due to the focus of the UK government around, they've put investments around mRNA and oligonucleotides. But I think what we'll find is the work that we're doing will be highly applicable then to other payloads. And then it's also about, again, if we come back to what the government's trying to do with catapult centres, it's also around kind of attracting inward investment, making sure that there are companies coming to the UK, that people are employing more people, that there are skills and knowledge transfer across the sector. So it's not just about the science and the research, it's about building an ecosystem and a support network. And just as an idea, this was what the funding agency Innovate UK said that they wanted to achieve. You know, we worked collaboratively with them on this. So you can see it's not just about the innovation, but integrating activities from research to commercialization. Hopefully you can see up there, there was a mention to international research. So despite some things that have happened over the last few years, there is a recognition within the UK government and funders that we have to keep collaborating with our partners internationally, or we will never be at the cutting edge of anything. So there is certainly, although some of these initiatives have a kind of strong focus on UK benefit and kind of economic benefit, that doesn't create a barrier to us ever working with companies or academic centres or research centres in Europe or beyond. And you'll also see a kind of a focus there on training and knowledge transfer within the community. So over the last kind of few years, we've been talking to both academics, but also industry, both large and small, and looking at what the challenges were that we might want to address. So some of those things that kind of came back, and we initially kind of created this, and it was very much focused on lipid nanoparticles, and that will be the lead technology. But we will be looking to apply some of the frameworks and assays and approaches to other delivery vehicles as well. So certainly there is something that keeps coming up again and again is around the analytical methods. So in vitro, in vivo correlation, how can we improve those methods? Having formulations that are thermostable and also suitable for alternative delivery routes. So yes, intravenous, intramuscular, but also intranasal, oral, transdermal patches we're starting to see. Understanding payload, transport and release. What do you change in that nanomedicine, in that delivery vehicle? What do you do with the chemistry and the structure? And how does that then impact on what it does once it gets into tissues and cells? Delivery beyond the liver, certainly with LNPs, comes up again and again, and the answer may be a different LNP chemistry, it may be a targeting molecule, it may be just not a lipid nanoparticle and use something else entirely. I'm sure we'll get to that. Predicting immunogenicity, safety, adverse events. Again, we have good methods, and a lot of them have come out of the European programs that we've heard about, but how can we take that a step further and integrate it into kind of high throughput screening? Something that comes up with companies and academics wanting to commercialize technology is unclear pathways to product registration. how do they get there, what do they need to do to get there. And so there seems to be a gap, or certainly a lack of kind of dissemination of information in that area. And then understanding all of the other stuff around how do you put a supply chain, including manufacturing, in place that will, create a stable route to market. And we're having novel materials here, so sometimes there isn't a GMP supplier for this new raw material. So we need to think about that. And then the complex IP landscape, particularly in the LNP space, the licensing conditions around certain lipids is a definite barrier to some people developing medicines in this space. So we've put together the work packages with those challenges in mind, and some of them are R&D based, putting together capability, developing enhanced methods, and some are kind of softer business support methods around what's the right framework to support a spin-out company from a university, how do we make sure they're getting the right advice at the right time. So this is the lead team. Hopefully you will see some familiar faces. Most of them are here over the conference. So I am and CPI are kind of leading the initiative, just making sure that we're managing and moving it forwards. But we're kind of very delighted that we've also got Yvonne Perry from the University of Strathclyde, Neil Littrop from Liverpool, you've heard from him a few times today, Robin Shattuck at Imperial College, and Sarah Brockbank at the Medicines Discovery Catapult. So we've kind of tried to bring a range of people together that both enables us to do the translational part, and what's quite interesting about the universities we're working with, they have also got a lot of experience of working directly with industry, but it also allows us to reach back, because there are still scientific challenges to solve in this area. So we need to be able to go back to that fundamental, the earlier TRL levels, and pull them through into this new IDDC centre. So some of what we're actually doing and what we've started to do. So some of what I wanted to do at CPI was lean on our expertise around robotics, high throughput processing, digitalization, AI modeling, which we do in kind of other areas and apply it to LNP screening in the first instance. So we're almost creating this lab of the future proof of concept using a collaborative robot that can work with a liquid handling robot that allows you to do screening of different formulations, which lets us look at certainly different ionizable lipids, but also the structural lipids, ratios, different kind of buffer formulations, excipients, and then move it into various cleanup operations and analytical characterization tests. And the ultimate aim of that is to link that workflow to an AI software that allows us to do directed optimization. So ultimately that feedback will tell the first liquid handling robot what it needs to do. We're a fair way away from that at the moment. We're trying to get the methods as robust as possible and trying to get the workflow sorted. But this is where we're aiming with this. If I introduce what that looks like in terms of a workflow. So yes, you've got something with an LNP like preparation of feed streams, and how do you, all of this you have to work out what's the best way to do it. How long is your lipids in ethanol stable for? Is it better to, have 4 different lipid stocks or mix them up for a particular experiment? And we were looking at both conventional pipetting and microfluidic technology, and then various reformatting on different 96 well or 386 well plates, that allow solvent removal, buffer exchange, dilution, sterile filtration, and then moving on to the characterization. So there's a lot of really cool characterization techniques we'd like to apply, but this initial part of the workflow is about high throughput. So if it doesn't work on a 96 well kind of format and is going to become a bottleneck, then we can't integrate it right now. One of the reasons for having a kind of collaborative robot that moves between, though, is things are developing at such a speed right now that we don't want it too fixed. We want to be able to plug in new technologies as they become available. So we're designing it in that kind of way from the start, because everything's moving so quickly. So we're working at the moment on making sure our encapsulation efficiency ribogreen assay is robust and giving us good results with different buffer starting, making sure we've got some measure of RNA integrity, because how do you know if it's the LNP that's falling to bits, or the payload that's falling to bits and hasn't stayed intact and it isn't having its effect. So it's quite important. We've got key things we need to measure along the way. So that would be CPI's parts, and just understanding why we want to go high throughput. So we think the current kind of bit of automation we're putting in should allow at least 8 times improvement in throughput, which starts to let you look at lipid libraries. I kind of was joking with some of our other academic leads, and they're saying, I don't know why you need that, you just get another postdoc. But one of the reasons for using robots is actually I'm as interested in the data set as a whole as the individual experiments, because once you've got kind of a repeatable automation to give you data, then you start to be able to do models and correlations of this chemical change or structural change, you know, is correlated to that effect. If you have kind of one person or several people doing experiments, and we're finding this in the early part of our work now, they just aren't repeatable. And so it's quite a lot of work to get it set up, but I think it will be worth it. So we're well on our way with automated preparations of LNPs, just kind of multi-channel pipetting on liquid handling robots, reformatting them into other plates for characterization like dynamic light scattering, and then moving on to machines for things like a ribogreen assay, which is coming along. We're getting pretty good results on that. And at the moment, the latest bit of work is, you can't see with the picture, but that's looking at tip geometries. So it's never going to be the same as a microfluidic manufacture of an LNP, kind of aspirating up and down in a tip. But you can look at kind of user engineering principles around the geometry of the tip, the speed that you're going out, the headspace, the pressure differential, to make it closer. And we're getting good results. We've already getting much more representative particle size and distributions. So there'll be more work in that kind of regard. Where this is, I'm not going to talk too much around these other areas because it's not my centre of expertise, but this, for instance, is what happens downstream of that once we start down selecting some of these formulations. So this is work that's happening at the Medicines Discovery Catapult, and where they really have the expertise is what happens and the mechanistic work of what happens once it gets into cells. So we have, you know, high content primary screens to look at biological activity. There's some really kind of cool high resolution microscopy and in vivo imaging that we can draw on as well. So that it's looking, you know, at both what's happening within the tissue, what's happening at the cell level, and then what the biological response is. So that gives us already a huge amount more data than if CPI were just working alone, where we would probably just start an on-off potency test. And then equally, if once we've kind of got through that, we also can draw on the expertise of other partners. So this is some of a summary of the work that the University of Liverpool are doing. So we've been able to benefit from the work that's happened in some of those other European projects. And so we already have access to a really good kind of analytical cascade around immunocompatibility, which can be targeted. Remember at the start I said it's really important to tailor this for things like route of administration, where you want to get to, but also kind of patient groups. So we have access to something already around here, which goes from kind of pre-screens when we send samples and ship them between partners, to acute toxicities using from whole blood responses, also all the way down to specific immune cells responses. So we already have access to a lot. We've been able to enhance the capability with some new equipment and platforms at Liverpool. But equally, it's not just about what's already available, that idea that we still want to be developing more. So there is a particular work package that is looking at how do we develop kind of better human cell models that will predict in vivo patient responses. And so that is in development with kind of some of our partners. And so they will be looking at how they develop new and enhanced methods, looking at things like predicting of biodistribution, potency, safety, and all of that. But we needed to pull that out, I think, as a specific work package because it was so important and the feedback came back so often. So I'm not going to talk through this in detail, but this is where we're trying to get to. Hopefully, some of you will be fairly familiar with that cycle around, it's rational design. It's thinking about what your target product profile is, critical quality attributes, how that's linked to manufacture and how you are manufacturing and controlling certain process parameters within that. So all of the work that we're doing around screening, taking it through characterization, there's also going to be at University of Strathclyde, they're going to be looking at some of the sort of manufacturing with different formulations and LNP chemistries, and all the time bringing that back into statistical modelling to understand those linkages. But the aim, why would we do that, is rational design, doing less experiments, using less material in the starting point, so that you don't have to do all of the formulations, all of the experiments. You could shortcut because you already have that database, that knowledge. And yes, we're starting with LNPs, but we will in phase 2 look at a range of other nano delivery systems. So we're kind of interested, and I have been talking to all of these types of different organizations over the last few years. A community, and a healthy, thriving community, is not just made up of drug developers. You need all of these different, the people developing manufacturing instruments, the people working on software packages, raw material suppliers who are interested in, converting things, GMP processes for new lipids or nanomaterials, CDMOs and CROs who want to develop services. For me, it's not competition if someone develops services similar to what we're doing. I think if we can help them do that, that's a success. We want there to be more companies working in this space, offering services, whether that's as manufacturer or research services. And so how can you get involved? And some of this is what you can do now. Some of it is what we're working towards in terms of operating models. So certainly you will be able to create a bespoke project that might be just a straight commercial fee-for-service piece of work. It's how catapults already operate, which will involve kind of usually more than one of those centres, depending on the scope of work. We're also looking at a membership model, which at this stage there's not enough results for it to be like a high-cost membership model. But at this stage it's more around working with a consortia to develop something that we call a grand challenge, which requires much more industry funding. So working together with government funding, industry funding, and leveraging the outputs of that academic research. Because it's been a 10 million investment over 3 years in primarily catapult centres and research technology organisations, universities. They are expecting that to lead to an investment of various companies in these projects. So there has to be follow-on work, whether it's commercial services or further collaborative programs where we get industry investment. And I mean, the next thing we're doing is an intracellular drug delivery task force. We had one earlier in the pandemic to create the business case, but really what I think of as next steps, the way funding cycles work in the UK is we got the money to do 10 million, get this initial investment. I need to be talking to government now on the next comprehensive spending review, saying what do they need to spend money on next in terms of business support, where do the research councils need to be prioritising their funding. So we've got a range of things we want to do around what funding fiscal measures will specifically support this sector, the technology roadmap. There are so many of you working on loads of different interesting technologies, but which are the short-term opportunities that are nearly there with a bit of a boost? Which ones are really exciting but probably need another 5-10 years of research funding in universities? So being able to kind of map that out for government, and also the regulatory kind of roadmap and pathways. It may be clear to some people who might have done it before. It's not clear to a lot of people who haven't taken a product to market before. And so how do we make that easier for them? And then a plan for knowledge transfer training skills. If this is going to be a large sector within the UK and globally, we need to make sure we're training the workforce, whether that's scientists in university or practical manufacturing skills at companies. So those are the 4 areas that we're hoping to get a wider view on from industry and academia so that we can then advise government with a single voice. I think that's all I had to say today. We're pretty early in development of this. We got the money at the start of the year and we only officially launched it probably last month. But would be very happy to talk to anyone over the conference if you'd like to find out more. Thank you.
Thank you very much, Juliana. We have time for 1 question, maybe 2, if there are some. Who wants? Yes.
Hi, Juliana. This is Katrina from MPL. I'm very interested in the use of AI for correlation of in vitro and in vivo. And my question is around where do you start and where do you stop collecting data and metadata? Because, you know, even the lipids, as you say, you know, how do I handle them or where they come from? You know, it's all information I need to build this correlation. So what is the rationale there?
So at the moment, we're working quite hard with our data scientists and IT colleagues to build some databases. So a range of databases, one with lipid information and chemistries. And, you know, some of the lipids we're looking at, there's, you know, not just 5 parameters. There might be like hundreds of parameters extracted from each of those lipids. So you can't put it in an Excel database. It's just going to break and destroy everything. So we're looking now at cloud-based databases. It kind of has to have the metadata in, but we're working out at the moment how much metadata for each of the partners and the type of work that they're doing you need to bring in. But there's nothing worse than getting that and then not knowing whether it was a different user or someone kind of changed something on their instrument, and that's why you're getting a certain result. So I think generally is use something that's capable of holding lots of information like a cloud-based proper database and collect as much data as you possibly can from the start because you can't go back and get it later. Once you've got that, 90% of the work with any AI or modeling is getting your data set clean and sorted and functional. Everything else beyond that is much, much easier. And I speak from experience because I did work on this.
Very inspiring. If you're interested in another way to use AI for nanomedicine, there's a very cool project that just ended, which is called Evo Nano. And they are more using AI for the design of the nanomedicine by itself. And it's great. I mean, I've learned a lot. Some key parameters you would think will have an impact are totally different when you put it in a big black box that thinks for you. So have a look at their results. They are published, Evo Nano. Fantastic. I mean, what a team, and you have the chance not to be European-wide, so you can work on a more tangible scale. We wish you the best for this initiative. Thank you so much, Juliana.
Thank you.
For the last talk, yeah. I'm really happy to welcome Lorna Fitzpatrick. Hi, Lorna.
Hi.
Different talk, you know why? Because Lorna has, submitted an abstract, and she's the only one selected for ETPN event. She's been reallocated because it was so translational. So we are super happy to have you here for the Medicine Discovery Catapult. And we're a bit late, but take your time.
Okay. Thank you.
Probably 15 minutes would be optimal.
Okay, so I can... Oh, sorry. No, that's all right. I can skip through a couple of my slides anyway to keep us on time. That's fine. Okay, so hi, good morning, well, good afternoon. And firstly, thank you to the organisers for inviting me to talk today. I'm really delighted to be here. So my name is Lorna Fitzpatrick, and I'm a lead scientist in cell and molecular engineering at the Medicines Discovery Catapult. And today, I'd like to spend some time to demonstrate how the MDC has positioned itself to support the industry to progress the development of new nanomedicines. So kind of a follow-on as from what Jules said. So who is MDC? So we are Medicines Discovery Catapult, established by Innovate UK in 2016 as part of a wider network of catapults. And we are an independent, not-for-profit organisation, and we're based in Alderley Park in the Northwest. And basically what our goal is to support the UK government's life sciences vision, which is a 10-year plan to push the UK to become a leading location of life sciences. So to do this, we've employed quite a large team of scientists with a really broad range of expertise across the life sciences, drug discovery and drug development. And how we work is that we work with SMEs, academics and larger pharma as well to help them push their therapeutic or their technology closer to the clinic. So we're really lucky. We've got amazing laboratory facilities, and that's really important for us so that we can support our virtual partners, so companies that don't have any labs at all, or our partners who have limited access to kind of cutting edge technology. So, as we all know, with the kind of last couple of years of everything that's gone on and the development of the coronavirus vaccines, we know that the nanomedicine space is a super exciting space at the moment, but translation to the clinic is still really difficult. So us as a Catapult wanted to figure out how we could help. So in 2021, ourselves plus CPI, who Jill just spoke for, we got together and we published a report called The State of the Discovery Nation, and that was based all around nanomedicines and trying to engage with UK stakeholders in the nanomedicines field to identify barriers that are difficult to overcome. So we identified 5 translational barriers. I won't go into the government investment and the regulatory process because we've kind of already discussed that. We know that that's difficult, particularly with government investment for small companies who are trying to generate proof of concept data to sort of boost investor confidence. It's really difficult for those to secure funding. But what I will talk about today are the kind of technical barriers that we identified, and that's kind of where we've come in to try to help. So the first one is that there was a real need for better access to drug development and formulation expertise, so understanding stability of nanomedicines or understanding the optimal conditions to combine a cargo with a platform. There's also a real need or drive or want for better access to preclinical imaging facilities to understand the fate of a nanomedicine in vitro and in vivo. And then also a requirement for better bioanalytical methods and validation assays to fully characterize nanomedicines. So again, I won't go into this for the sake of time in too much detail, but in response to that report, what MDC did was we developed capabilities across 4 key areas to try to help the sector. So firstly, we have a virtual R&D team set up, and the goal of that team is to help collaborators evaluate their critical data, generate a roadmap that includes key milestones to help transition into the clinic. We also have a really wide network of expert partners and CROs, and where we feel we can't give the help or guidance, we lean on these for support, and we're more than happy to link up our collaborators with our expert partners. In terms of the wet lab, so we have set up advanced imaging technologies to understand physical characterization, and then in vitro and ex vivo, we've set up a number of assays predominantly focused around microscopy, looking at things like cell-specific binding, internalization, trafficking through the endosomal compartments. And then also we're really keen, and what we're doing at the moment is trying to optimize better imaging methods to be able to track co-localization of your delivery platform with your cargo. In terms of preclinical imaging, we offer bespoke radio labeling and fluorescent labeling of nanoparticles so that we can track biodistribution, as well as then being able to sort of predict efficacy a little bit better. And then finally, we've got a biomarkers team who can do analysis, all the sort of similar analysis that we do in our cellular assays, but doing this ex vivo to look at tissue penetration or tumor penetration, accumulation. So all the same type of stuff that we do in vitro. So another theme that came out quite strongly in our State of the Discovery Nation report was collaboration. So over 2/3 of the respondents actually said that they wanted more support for collaborative partnerships to increase their knowledge and expand their expertise. And us as a Catapult, who are relatively new to this nanomedicines field, agree, and we're really, really keen to collaborate as well. So we collaborated with Seda Pharmaceutical Development Services, who are a company that are also based in the Northwest, who have a lot of knowledge around formulation, nanoparticle design, development, and the regulatory requirements as well, and have seen products through to commercialization. So we set up a collaboration with them so that we could kind of combine our area of skills in characterizing nanomedicines with their formulation expertise, in the hope of together, you know, we would be in a better position to positively impact on the sector. So for this project, what we did was we firstly wanted to develop 2 types of nanoparticles and develop them in a slightly different way of processing, and try to understand if we were able to implement our assays to see if the change in process would have any impact. So what we first did was we took sRNA and we labeled it at MDC. We then shipped this to SEDA, who developed both lipoplexes and LMPs. So for the development of lipoplexes, what was done was lipids were added into ethanol. The lipids were allowed to form, and then we added the nucleic acid afterwards for complexation. However, in terms of the LMPs, the sRNA was added at the same time as the lipids, and complexation was allowed to happen. And we made an assumption that the sRNA was condensed by the ionizable lipids into inner micelles within the LMPs. So what we then did, well, at SEDA, they did some physical characterization. So what you can see on the top right is that when we did size distribution by DLS, both particles looked pretty similar. And then similarly for zeta potential and encapsulation efficiency by ribogreen, we saw very similar results for both the lipoplexes and the LMPs. So next then, SEDA also characterized by looking at stability. So looked at stability at 2 to 8 degrees over 7 days. And what we found, again, the LMPs and lipoplexes were similarly stable in terms of size and polydispersity. The particles then were shipped to MDC, where we were able to treat a cell line that we had previously genetically modified to include a GFP tag on Rab7, which is a late endosomal marker. And what we did was we treated our cells with both the lipoplexes and LMPs, and then looked at uptake at fixed time points. And what we can see is in the graph on the right, we see total accumulation, and it seems to be maximal accumulation about 8 hours. And we see a corresponding level of accumulation or co-localization with the Rab7 at the same time point. So you can see there that the sRNA, which is what we were measuring, is labeled in white. So again, it looks like both particles are performing pretty well in vitro. Next then, what we want to do is see were both of these particles capable of endosomal escape. So we really subtly modified the protocol that was published by Munson et al. In 2021. And what we did was generated a lentiviral construct that fuses galectin-9 to GFP. So in normal or healthy or untreated circumstances, you can see on your far left, that GFP, Gal9, is quite diffuse throughout the cell, and when you treat the cells with both lipoplexes and LMPs, what we can see is spot formation. And we're actually able to image this live over time and count the number of spots. So again, similarly, with the uptake, we see very similar levels of endosomal escape, which kind of comes on around about an hour. We start to be able to see spec formation. So then finally what we wanted to do was understand if we were actually able to deliver sRNA into the cytoplasm that was functional. So prior to us shipping the sRNA to SEDA, what we did was we labeled it up, and we just wanted to do a Western blot and make sure it was still functional. So the sRNA that we're using in this study is one that we've used previously. It's to a protein that is expressed in many cells, the histone methyltransferase called EHMT2. And what you can see in the Western blot on the left-hand side is that at 48 hours we see pretty similar levels of knockdown with treated or with labeled and unlabeled sRNA. So we're happy with that. We shipped that over to SEDA. They encapsulated it, came back to us, and then we looked at knockdown efficacy of both the lipoplexes and the LMPs. And once again, we saw very similar levels of knockdown at this one particular concentration that we chose, which was based on the literature. So one could say from all of this data that we've generated, it looks like the lipoplexes and the LMPs behave pretty much the same way. So I guess if you're in a situation maybe where you had a collaborator doing this, you say, okay, well maybe, we could go in vivo now with this and potentially it should, act in the same way. However, when we then decided we really wanted to understand, are these particles actually different? So what we did then was we employed super resolution microscopy, and what you can see in the top lipoplex there is due to the small changes that we made in formulation. It's really obvious to see by super res the sRNA quite nicely around the lipids, whereas with the LNPs you can see that the sRNA is quite dispersed through the LNPs, which would suggest potentially better encapsulation. So I think if you were to see, looking at this scenario, you would maybe be slightly bit more reluctant to go in vivo, say, with a lipoplex, just because you can see that your sRNA is so exposed. So even in an in vitro situation, these both perform the same. I think it's quite important to do this extra, or as much characterization as possible, to really know kind of what you're dealing with before you start moving in vivo. And then just finally on the right-hand side, we're also able to do size distribution using super resolution. We got very similar results to the DLS. But the quite nice thing about the super resolution is you can see a tiny little tail at the end, which potentially would suggest a little bit of aggregation, and that's something that you don't quite pick up as well with the DLS. And the super res is quite nice as well because your particles don't necessarily have to be spherical for the analysis. So hopefully that project kind of gives you an idea of the types of assays that we can do in vitro to try to help characterize a nanoparticle. So then in terms of in vivo, I want to just go through one case study that we've done where we have worked with a small SME and have had quite a big impact on that small SME by doing some biodistribution and efficacy in vivo. So we're super lucky at MDC. We've got a really fantastic translational imaging capability, and we're implementing this more and more in our nanomedicines projects. So we have the capacity to do optical imaging, so bioluminescence and fluorescence, ultrasound, PET and CT. And to date, we have successfully both radio-labeled and fluorescently labeled delivery systems, as well as cargo, such as sRNA and antisense oligonucleotides. And the way in which we work is we'll always work with our partners, discuss with them what the best method of imaging might be, because they all have their own advantages and limitations. But of course, we'll take into account the characteristics of a nanoparticle before we kind of make a plan on how we're going to do the imaging. And then also, we have excellent radiochemistry capabilities at MDC, and we often employ these in our biodistribution studies just because of the sensitivity we get with radiolabeling and PET. So in 2021, MDC acquired the Wolfson Molecular Imaging Centre, which is the radiochemistry facility in the University of Manchester, which was actually shut down in 2020. So we've relaunched it, and it's a dedicated facility for PET imaging and radiochemistry, and it just allows us to develop novel radiochemistries that we can employ then in labelling nanoparticles for biodistribution. So on the right-hand side of the graph, this is just an example of how we would go about labelling LNPs with zirconium-89 for a biodistribution study. So in the middle, what you see is we're able to do surface labelling, and on the right-hand side then you can see that we're also able to encapsulate the zirconium within the LNP. And also sometimes when we suggest these kind of studies, our collaborators might be a little bit cagey about it just because of the worry that adding on these kind of labels may alter the integrity of the LNP. But actually we can test these things in vitro cold before going hot in vivo. So we can do a certain number of tests to make sure we maintain the integrity of the LNP. So then this is one of our case studies that we worked on, and this shows one project where an SME were really keen to develop their nucleic acid-based nanoparticle, and they wanted to modify it so that it was tumour targeting. So what they did was they took their nanoparticle, added on a tumour targeting ligand, and then we labelled both the targeted, the non-targeted, and then just naked sRNA with zirconium-89. And basically what you can see on the left-hand side is we did PET biodistribution, and the main take-home message was with the targeted and non-targeted, we didn't see any difference in the levels of radio tracer within the tumour. So we took the organs ex vivo, and you can see here up on the very top right-hand side, there's clearly no difference in tumour accumulation with targeted and non-targeted, or even compared to sRNA. So although that was disappointing for the SME, it was really important for them to know this so that they could go away and redesign their strategy. And alongside this then, they're also just able to see biodistribution of their particle. So like many particles, we can see that we get accumulation in the liver and the spleen. So then what we did was we did a follow-on efficacy study where we switched out the PET radio label for a Cy3 label on the sRNA. And as we knew from the biodistribution, we saw accumulation in the liver. So we took out the liver, sliced and diced it, and what you can see in the image on the top right is that we got accumulation of Cy3 sRNA, predominantly in the Kupffer cells, over the hepatic stellate cells, endothelial cells, and the hepatocytes. So it's quite nice to be able to see this granularity. And then what we wanted to do then was look at, okay, so we have accumulation within cells within a tissue, but is there any functional effect of the sRNA? So in the bottom panel then, what you can see is the target that the sRNA was hitting. It was a constitutively active or constitutively expressed protein. And in the vehicle, you can quite clearly see that we get quite a lot of staining. The LMPs, which were used as a positive control, completely wiped out the target, and the nanoparticle was somewhere in between. So this was a really nice study for this relatively young SME. They understood biodistribution. They were able to show that they can get accumulation, penetration, and delivery into cells, which is pretty fantastic for them, even if the nanoparticle didn't perform quite as well as the LMP. So that kind of gives you a flavour of the type of work that we do at MDC, and in the future we're dead keen to continue to collaborate and increase our impact in the community. We like to listen to the innovators in the field and try to be innovative and versatile and flexible in the types of capabilities that we build. So yeah, that's it. Thank you for listening, and sorry if I have gone over.
Thank you so much. We have time for just one quick question, if there is. The first one to raise their hand. Oh, yeah, please.
Very, very quick,
Yeah.
really quick. And thank you so much for sharing. I want to ask the following. Other commercial CROs, they don't share data with, findings. You have shown us today distribution, and that was very, very interesting for us, that we're doing distribution kinetics, how much tumors take up, etc. Is there a chance that when we come to you that you can share some information from the other SMEs with us?
Yeah.
I mean, just to improve the next steps,
Yeah.
because it was really impressive what you did today.
Yeah.
Thank you.
Yeah, for sure. So it depends on the type of project that we work on. So some of the work that we do will be fee for service, so we won't necessarily own the data. But for these were grant. We were partnered on grants, so we also own the data, and definitely then in that case we can share what we've learned. So
Thank you.
Thank you.
Thank you so much. I won't take too much of your time. We're already late. Just 3 minutes, because I thought that would be a pity not to mention that. We've talked about, you remember the nanomedicine translation hub with the UNCL and the OETBs, etc. I just wanted to mention the health tech tab, because it's still alive, and I guess it's still unique in Europe. So if you guys are doing translational nanomedicine and looking for industry expertise to transform a good innovation in a possible solution, a credible solution for patients, you should have a look at the health tech tab. We'll go super fast. This is you remember Maya, the article on TRL? I found it. It's this one, just to let you know. Oh, she went away. No, basically just the tab to let you know it was part of this big picture. The tab for the mentoring, the UNCL to help on characterization, and pilot lines, and then OETBs for the manufacturing and a more global point of view. And just in one slide, so it's basically about people, the tab. We have within this board, this translation advisory board, one of the, let's say, top players of the game, ex-people from Bayer, from Siemens, from Innovate UK, from Ipsen, from Medtronic, and also entrepreneurs of the nanomedicine game, like Indeed, Eric, Cécile, and Laurent, that you might all know very well. And what they do is just help you asking yourself the right question. Are we addressing real clinical needs, not just the one we pretend to address? What is the real application we should go for? Is there a real market? What is the real competition, which is not the same people, other people doing the same technology, but people developing an alternative solution that can be 10 times cheaper than yours? So asking the right question basically make academia think like an SME and making SME think like industry. We have supported more than 150 projects for 7 years. with some success stories, some testimonials I would be happy to share. Just to let you know, the EU funding for the tab is over, like for the UNCL, like for OETBs. But we work now on a little service for fee. It's open for applications. We'll do a big session this year. This is why I just wanted to say it's still alive. And it's unique in Europe, not because the advice is good, because it's peer-reviewed between the experts. It's the only group of peer-reviewed mentoring, custom-made for your needs by industry experts in Europe. It does not exist at the EIC. It does not exist within EIT Health. It does not exist at the IHI level yet. So just to let you know, for instance, we had a client for the first time, not just a beneficiary, people from Sintef that did a spin-off after the help of the tab last year and just launched this company Nadeno that we are happy to see alive. And yeah, proven value. Just have a look at the website. If you don't find the information you want to have, just contact me. I'm easy to reach, and I'd be happy to push you to the experts. You're thinking about translational, you don't know how, you don't know if it's the right application. These are the best people you can find in Europe to help you doing that at, it's not even cheap, it's ridiculous cost. Just wanted to mention that. So the tab is alive. Yeah, it still moves. Thank you so much. It was a great session about translational medicine and nanomedicine. Now we have the lunch break, and we'll be back at 2 in Lecture Theatre 1 for a plenary session. For a short sponsor talk and then a plenary session organized by our dear partners from Elrig UK. It will be fantastic. We have a great program by the afternoon. Have a great lunch.